Welcome to the 8th Annual Seattle Area.
The Seattle Epidemiologic Research and Information Center, in collaboration with the Department of Veterans Affairs, the VA Employee Education System, and the University of Washington Departments of Epidemiology and Biostatistics, presents the 8th Annual Epidemiology, Biostatistics, and Clinical Research Methods Summer Session.
Good morning, everyone. I'd like to welcome you to the 8th Annual Seattle ERIC Epidemiology, Biostatistics, and Clinical Research Methods Summer Session. My name is Ed Boyko. I'm the course director. Today's course title is Genetic Epidemiology, and I'm pleased to introduce to you today's speaker, Dr. Karen Edwards. Dr. Edwards is an associate professor in the Department of Epidemiology and the Institute for Public Health Genetics, and director of the Center for Genomics and Public Health at the University of Washington. And the title of our talk today is Segregation Analysis. Dr. Edwards.
Good morning, everybody. Today we're going to cover segregation analysis, which is another approach that we can use, again, to think about our phenotype. And in this case, what we're actually doing is modeling different traits to see whether or not they are inherited in a Mendelian fashion, and now we're using family data. So in this particular approach, what we're doing, again, is looking to see whether or not there's evidence for genetic influences on a particular trait, and we're now back to thinking about single major genes that underlie the trait. So what we're doing in a segregation analysis is using our family data, looking for co-segregation of phenotypic traits through families, and then using that information to try and model the mode of inheritance. So for example, is a particular trait, is the inheritance pattern consistent with dominance effects, recessive effects, or codominance? And again, we're thinking about single major genes now. Complex segregation analysis relies on the concepts of Mendelian transmission and Mendelian inheritance, which, if you think back to the classic experiments with peas, this is what we're talking about. So complex segregation analysis is actually quite useful in not only thinking about your phenotype and trying to determine the mode of inheritance in families, but the information that you gain from segregation analysis can also be quite useful when you then move on to do linkage analyses. And in particular, when you're doing parametric or lot-based linkage analyses, you need to specify certain parameters in your genetic model which you can get from segregation analysis. So segregation analysis used to be something that genetic epidemiologists spent a lot of time doing before they ever moved on to doing linkage analyses. However, currently, most people do not do segregation analyses. They just jump right into the linkage analysis. Part of the reason for that is, in the old days, let's say 10 years ago or so, it took a long time to get genetic data, and it was very expensive to get. So frequently, you would have years while you were waiting for your genetic data to come in. Segregation analysis is actually quite time-consuming to do, so you could fill those years running models and getting ready for that linkage and genetic data. Now we get that data so quickly, you wouldn't even have time to run one of these genetic models in a segregation analysis. But people still do it occasionally. You may still come across these in the literature, and there may be some circumstances in which you may want to consider doing a segregation analysis yourself if you've got the family data. So what we'll do today is go through the basic concepts of segregation analysis. I'll show you some models. We'll look at the parameters and how to interpret these. As with many of the methods that we use in genetic epidemiology, there is an art to doing segregation analysis and interpreting the results from such an analysis. And again, one of the reasons why we spend time in this course talking about segregation analysis is it's likely that you may run across these in the literature. There actually have been quite a number of segregation analyses published, and these can be quite useful in helping you, again, think about your phenotype, which traits may be more amenable to doing linkage analyses on and actually looking for genes for particular traits. So you will not come out of this class actually being able to run a segregation analysis, but you'll be able to look at them and interpret the results. We could spend quite a long time running segregation models in here and demonstrating that software. So what we're doing in segregation analysis is looking for compatibility with Mendelian expectations of inheritance and estimating several different parameters that we use to construct genetic and non-genetic models. And then we're using maximum likelihood approaches to estimate those parameters, to estimate the likelihood, and then to compare nested models. So this is a modeling approach. We have no genotypic data here. We're relying solely on phenotypic data and looking for the co-segregation of phenotypes in our families. There are many assumptions that go into segregation analysis, and we'll talk about these. Some of the assumptions include that you have your pedigrees accurately defined and you actually know the biological relationships. And we'll talk about that and the importance of that in a little while. With most of the methods we've talked about so far, complex segregation analysis provides, if you will, in a sense, statistical evidence for a single underlying gene. Again, it's not proof because we don't have any genotypic data here. We haven't actually identified any genes, but we're adding to the evidence that the trait is under genetic control, and in this case, more specifically, whether or not it follows a particular mode of inheritance in our families. The other thing that is important about segregation analysis is, in many cases, we can also explore heterogeneity using our family data. What we may find is that in particular families, we have evidence for co-segregation with a particular trait, but in other families, there's not. And that would be an indication that there's heterogeneity, and it may be that you want to select a subset of your families then for future studies where you do the linkage analyses. If a trait's not segregating in a family or doesn't appear to be under genetic control in a particular set of families, then you may want to exclude those families and not look for genes in that particular subset. So segregation analysis, even though, as I said, it's not done as often as it used to be, can still be helpful and can still help you think about your next steps in your studies and hopefully make you a little more successful in finding the genes. Now, there are several different software packages that can be used to run segregation analyses, and most of these simultaneously consider the effects of a single major gene, polygenic component, and in most cases, environmental factors as well. Most of these packages will allow you to include environmental covariates in your models. So the approach in general is to fit a series of models to your family data and then compare these nested models using a likelihood ratio test. And what's interesting with the segregation analysis is when we are testing these models, it's a slightly different approach than what we use in normal statistical approaches. Here we're looking really for a goodness of fit, and the null hypothesis is that the data do fit with some model of inheritance, genetic or not, which is important. And this is when we start talking about the interpretation and selection of the best fitting model, it gets a little bit more confusing than what we're normally used to thinking about. Okay. So the models, and I spend a fair amount of time getting you ready to look at the results from a segregation analysis because the first time people see these, it's a little overwhelming and they think we can't possibly understand all these different parameters. So we're going to talk about them first, and you'll see that there really aren't that many parameters that are used in the model. We'll go through each of the different parameters, talk about what it's doing and how it's estimated and what the implications are for those particular parameters. Now, this should sound in many ways similar to a commingling analysis, but it's being extended and we're using family data to estimate many of the same parameters. So some of the concepts should be similar and sound familiar to you. So with the segregation analysis, we have what we call our most general model, which is where all the different parameters are estimated. And that traditionally is the model which others are compared to, because then every model is nested within the general model. The different types of parameters in the model relate to the single major gene component. So what we try to do with a segregation analysis is build models that allow us to choose from different genetic models versus different environmental models, with the idea that at the end of this, we end up with one best fitting model, which either tells us that there's a single major gene or not, and if there is a single major gene, how is it inherited? So in doing this, we fit a series of different models. One type of model that we're going to fit is a model that relates to estimating just the single major gene components. Okay? And we assume in some of those models that there's no polygenic inheritance. So what we're saying is that the variation in the trait is due entirely to a single major gene with no additional polygenic effects. We can also estimate a pure polygenic model where we assume that all the genetic effects are polygenic and there is no single major gene. We can estimate mixed models where we assume there are both things. There's a single major gene and then there's additional residual polygenic effects. And then this is a relatively newer introduction into segregation analysis, is the incorporation of non-genetic models. Because in the early days, people only ran genetic models. And so by default, your best fitting model is going to be a genetic model. So it's very important to also fit a non-genetic model and make sure that that does not fit your data better than your genetic alternatives. So with segregation analyses, we fit both genetic and non-genetic models. We fit a variety of different genetic models to try and figure out which is the best fitting model given our family data. Okay? So we'll go through the parameters here, keeping in mind that the two types of genetic effects that we have in our models are single major gene effects and then the polygenic inheritance. Okay? So we'll start first with the parameters that relate to the single locus component or the single major gene. Just like with commingling analysis, if we have a single major gene, and let's assume that we have two alleles at that locus, we remember that each genotypic class is represented by a subdistribution, and each of those different subdistributions has a mean, a standard deviation, and a proportion of individuals that underlie that distribution. In this case, we're estimating similar parameters. We're going to estimate the mean for each of those subdistributions, which corresponds to these theoretical genotypic classes. We're going to estimate the variance for each of those subdistributions, and we're going to assume a common variance like we did previously. And then we're also going to estimate allele frequencies now, rather than the proportion of individuals falling into each of those subdistributions. So now we're actually starting to estimate allele frequencies, even though we have no genotypic data. We're modeling these things. The parameters that will probably sound most foreign to you are what we call the transmission probabilities, or tau. And these we assume relate to Mendelian expectations. And I'll go through these because they're a little confusing at first. We have no genotypic data, but we are assuming and we're modeling a single major gene that underlies the distribution in our trait. And what we assume is that if a parent has the big A genotype, the probability that they're going to transmit a big A to their offspring is 1, because that's the only thing they have to transmit, so the probability is 1. That's referred to as tau 1. Tau 2, if the parents are big A, little a, so a heterozygote parent transmits the big A allele to the offspring, should be 0.5, okay, because it's half that they transmit the big A, half that they transmit the little a. So that tau 2 is 0.5. Tau 3 is the probability that a little a parent transmits the big A to their offspring. That probability is 0. So these various parameters that I have up here, the means, the variance, the allele frequencies, and the taus, all relate to the single major gene component of our model. When we start building these models, these are the parameters that we have to work with to build the different genetic models that relate to the single major gene. So we're going to use this set of parameters to model dominant, recessive, codominant modes of inheritance in our pedigrees by manipulating these parameters that we see here. Okay? Then, as I said, we can also build mixed models or models that just have the pure polygenic component. And this parameter should also be familiar to you. This would be the heritability. And this is, as you heard earlier, heritability tells you about the proportion of variance that's attributable to additive genetic effects. It's not the single gene component, it's that polygenic component, where we cannot necessarily separate any individual single gene. Because most of the complex diseases that we're interested in are likely to be influenced by both single major genes and then additional genes, the polygenic component in many cases represents the genetic component, the residual genetic component that's left over after we estimate the single major gene effects. So it's not surprising in many cases that a mixed model fits our data better than just a single major gene model, particularly for complex diseases. Before I put up the segregation analysis result, there's something else I wanted to talk about. Whenever we're doing genetic studies, and particularly when we have family data or data on related individuals, it gets much more complicated to do any of the analyses than what we would need to deal with in a traditional case control type of study. And it's critical that you, when you're doing these analyses, that you correctly specify the relationships between individuals, especially when you have family data. Most of the software that you're going to use for genetic analyses, so things like SAGE that we talked about previously, software that we use for segregation analysis, actually require more than one data file to do the analyses. So you're used to having a data file that has all your data in it for your traditional analyses, but in genetic studies, we typically have to have several different files. One of the files that's required is what we call a pedigree structure file. So it tells the programs how your individuals are related to each other. The most critical pieces of information in these pedigree structure files are parents, because the way individuals are linked to each other is through the parents. So let's say you were to do an analysis and you only had siblings. If you just put two individual siblings in the data set, the file or the program would not know that those two siblings are related. So what you have to do is put in parents, whether or not you have data on the parents that link those siblings together. So when you're working with genetic data and family data in particular, you have to include parent IDs for every individual in your sample so that they can be linked to the rest of the pedigree. So it creates, in many cases, quite large files, even though you may not have phenotypic, genotypic data on those parents. They have to be included in the file to link your people together. And this is an example of a file that we use, a family structure file. And there's a particular format which, surprisingly enough, genetic epidemiologists have adopted so that across a variety of software programs, they all expect this same format. And it's what we call linkage format. And I'm going to tell you a little bit about this because if you're ever reading these software manuals, as I said, the documentation may not be that good, but they may say we require the file to be in linkage format, and that may be all they say. So I'm going to tell you what linkage format is, okay? Because it's pretty common across these different packages. So what we generally have is, particularly when we have family data, what most people do is each family will have an ID or some sort of identifier that everybody in the family shares. So there's a family ID. And then within the family, each individual will have their own ID. So the way that we've traditionally done it in our studies is the family ID may be a 6-digit number or may include some letters depending on how you do it. And then within that family ID, each individual has their own ID. And what we tend to do is make the probands 001. So the proband is the individual who brings the family to your attention. Maybe it's somebody that was seen in clinic, maybe it's a case, somebody who had a particular disease, and then you bring the rest of the family in to study them. This way we can easily identify who the probands are in our data analyses, and frequently we're wanting to look at those in particular or adjust for certain things, and so it's easy then to pull out the proband from each family. So we have the family ID, the individual ID, and then you also have to have, as I said, the father ID and the mother ID, and they have to be in that order. Okay? So father ID, mother ID, and then the sex of the individual. And then here we have our phenotypic data on 5 different traits. And this is what linkage format is. It's essentially the family ID, the individual ID, most importantly, the father ID, the mother ID, and the sex of the individual. Okay? So that's linkage format. As I said, when we start doing genetic analyses with related individuals, we're moving into much more complicated analyses where we have to have these additional files that define the structure of the pedigrees. Okay. Now, for segregation analysis, and many of the things that we've talked about previous to this, we have no genotypic data. And so we're not actually able to verify the relationships as we note them here. Frequently, when you're collecting this type of family data, you're asking individuals, who's your mother, who's your father, who are your siblings, and we're assuming that we're getting the correct biological information. It's surprising to people who work with family data to find out that the rate of non-paternities, I hate to bias it and say that it's the non-paternities, but it's the way we refer to it. We can refer to it as non-Mendelian inconsistencies. How's that? To be more correct. That we see that on average, about 10% of our samples are not related in the way that either the family thinks they are or somebody in the family thinks they are. So you can imagine when you start doing linkage analyses, if you have people in the family who are not related in the way that they have been represented by the family, it can cause some big problems in your linkage analyses. Okay? And we'll talk about that a little bit more, but the rate generally across a sample of families is about 10%. Now some of that could be lab error, sample mix-ups, and then the rest, other things. Okay? But it's really important to keep in mind. And before you actually start doing linkage analyses and things like that with the family data, most people spend quite a large amount of time cleaning their data and checking for correct relationships. And we don't have time to do that in this class, but in many cases it can take 6 months to a year to, once you get the genotypic data, check to make sure that everybody is consistent and related in the way that you think they are. And then you have to make some tough decisions about what to do with people who are not related the way that they say they are. Do you exclude them from your analyses? Do you exclude them from your database completely? What do you do with them? But it's a critical step as you start moving into working with family data, and one that if you ignore it, you'll have some problems down the road. Okay? So that's something to think about with family data. And with segregation analysis, we will assume that we've got our data, it's correct to the extent that we can correct it at this point. It may be that we don't have any genotypic data yet, so we're having to rely on the reported biological relationships. The good thing is with segregation analysis is it appears to be fairly robust to these Mendelian inconsistencies, unlike linkage analysis. So if you don't have genotypic data yet and you're just working with your family data, you're probably okay for your segregation analysis, but you certainly would want to check this before you go on to do linkage. So this is a fairly simple complex segregation analysis that has been reported in the literature. And I chose this one because it doesn't have too many models reported, and it also has the basic models that we would like to see. So in case you can't read this, here we have the general model. This is our environmental model or non-genetic model. We have a mixed model, mixed codominant. We have our major gene model. This says mixed recombinant, but I actually think they mean mixed recessive model. Mixed dominant model, polygenic model, and then what we call a sporadic model. And I'll go through all these again, and then we'll go through the parameters in detail. But what we have here in terms of the parameters are the three means, mu1, mu2, and mu3. We have P, which is the allele frequency. We have the heritability. We have the variance, so a common variance for each of the subdistributions. These all relate to, well, except the heritability, those all relate to the major gene component. And then we have the taus, tau1, tau2, and tau3, which also relate to the major gene component. So these are our transmission probabilities. Then what we also have that's important is the likelihood, from the maximum likelihood estimates that we're going to use to compare nested models. So a couple of comments before we get into actually looking at the model. When people run segregation analyses, everybody makes up their own labels for the models. And there really is not a lot of consistency in what people name models. So it's critical when you're looking at the results of a segregation analysis to look at the label, but then look to see what parameters have been estimated and which parameters have not, to really get a sense of what that model is. I mean, everybody's got their own system, and it differs from investigator to investigator. So although one person might call this model a general model, you may have a very different general model in another segregation analysis. Usually what you see is the model that's termed the general model is just the most general model. It's not necessarily one where they're able to estimate all the parameters, but it's the one that has the most number of parameters estimated. So of the set of models that they've built, it is the most general one. And this is actually, seems like a subtle detail, but it's actually important when you start comparing other models and looking at that general model, what they were able to estimate. Now I keep referring to this, what they're able to estimate. When you're running a segregation analysis, what you're doing ideally, is estimating all of these parameters simultaneously. So with the commingling analysis, remember we just estimated the means, a standard deviation, and the proportions in unrelated individuals. So pretty clean, pretty easy to do. Here we're now having to model not only those parameters, but transmission probabilities, heritability, and allele frequencies using family data where the structure of the families is much more complicated. So the ability to estimate all of these parameters simultaneously, it's very difficult to do this. You would never be able to sit down with your fresh data and run a general model where you estimate all those parameters simultaneously. Chances are that thing is going to crash probably in the middle of the night after it's been running for many, many hours, and then you come back in the next day and have to start it all over again. Okay? It is just too difficult to get good estimates of all these parameters right up front. So when you actually are doing segregation analyses, you start with the simplest model and build up. So your general model is usually the very last one that you run, which is a little maddening because what you want to be doing as you're going along, being the curious people that we are, is be testing those models, at least in your mind, to see how things are doing. Because you're sitting there for hours watching these run. You need to have something to keep yourself amused. But what we do is we start with the most basic, simplest model where we're estimating the fewest number of parameters. We get good estimates for those parameters, and then we build up to the next model. Okay? And there's a lot of back and forth here because as you get a better estimate for a particular parameter, then the other parameters start changing as well. So this is why segregation analysis can take literally months to get the results that you see here, because you've run many models, and then you also have to run each model more than once to make sure that you've got the maximum likelihood value and that you've got the best parameter estimate. Okay? So this is actually quite a time-consuming type of analysis. Okay, so let's look at our models, and let's look at the parameters that are in the models and how we construct these different types of models. So let's start with the general model and look at that. In this case, what we've estimated are the three means. So assuming that we have a single major gene with two alleles, each of those genotypic classes are going to represent a different subdistribution. And these are the means of the three different subdistributions, which I know you can't read, but there they are. You also can't see this, but the means are ordered. So for example, we have mu1, which we would assume relates to a homozygote class of 83. Mu2 is 127, and mu3 is 186. So there actually appears to be three separate means here. Just thinking about this, if this is what you saw right off the bat, what might you think about the mode of inheritance here? That's right, codominant. It's not unusual to see two of these means being very close to each other. So for example, it's possible that that heterozygote mean is closer to either mu1 or mu3. If that were the case, what might you think? Now, it could be dominant or it could be recessive, depending on what mu2 was closest to. So you can just tell by looking at these means, in some cases, what your mode of inheritance is likely to be. And that then would influence what other models you would build to test that. Okay? The idea behind your general model is this is the most general model, which you can then use to compare other models to. Okay, so in this case, the means are ordered. It appears that we've got three distinct subdistributions. Two of them are not overlapping, which would give us a different sense of mode of inheritance. Then we have P, which is the allele frequency. And again, this is arbitrary because we don't have any genotypic data. But we're saying if there is this underlying gene with two alleles, this is the frequency of one of those alleles. Because there are only two, they have to add up to 100%. So we've got the allele frequency here, one of the arbitrary alleles of 0.8, which would mean that the less frequent allele is going to be about 0.2, which is pretty common actually. Okay? We're not talking about very, very rare alleles. And then we have a heritability of 51%. Okay? So this is a general model, has both major gene and a polygenic component. And you can see that that polygenic component is actually pretty high. Given that you also are modeling a single major gene, it's saying that there are additional residual genetic effects contributing to that trait. And then we have our common standard deviation, and then we have the taus. And what you will commonly see in these genetic models is that the taus are very difficult to estimate. Remember we said for tau 1, the probability of tau 1 should be 1, which is really on a boundary. Tau 3 is also 0, which is on a boundary. You can only estimate it in one direction. There's not anything on the other side of it. So both of these parameters can cause problems in estimating because they're on the boundaries. Tau 2, which is, we would expect it to be about 0.5, we can do a little bit better with because you can estimate that from either direction. I mean, it can, essentially you can estimate anywhere from 0 to 1, although you hope it's 0.5. So it's not unusual for people not to be able to estimate these particular parameters. And in segregation analysis, when something is not estimated, we generally put brackets around it or somehow indicate that that parameter was held at a constant value and not estimated. And it's important to look at these results and see, did they estimate these parameters or did they set them at their expectations and not test them? Okay, those are the things that you're going to want to look for. In this case, they were able to estimate all these parameters, and then they have their likelihood value, which is printed here. Okay, then the next model you see on here is what they're referring to as the major environmental model. This is where the labeling sometimes is helpful, sometimes is not. But what a major environmental model in this case refers to is that the component that relates to the single major gene, what they're saying is it's not genetic, it's due to environmental influences. But they are allowing for a polygenic effect in here. So it's not a non-genetic model, it's just that the major gene component, they're saying, is due to environmental factors, but they're allowing for a polygenic component. That's actually important. If you're not able to build a purely environmental model, because sometimes these things just will not fit the data, that's usually a clue that that doesn't fit. So for example, if you could not fit a model where the major gene component was due to environmental effects and the heritability was equal to 0, that probably tells you the reason that model is not fitting is that there are genetic effects. You can't fit a non-genetic model to the data, okay, which is important. And that's probably what's going on here, is they probably could not fit this model without including some genetic component. In this case, it's the polygenic or the heritability. The other clue that we have here is that when you look from the general model to the major environmental model, look what happens to the heritability. It goes from 0.5 to 0.8. Anybody guess why that might be? What we've done here is basically tried to get rid of the major gene component. So if there truly are genetic effects, what's probably happening is those genetic effects are now subsumed in this heritability estimate. That's why it goes up, okay? Because there's no other place for the genetic effects to go. So again, that's an important clue. They then go on to fit a series of other models, which what they're doing now is just manipulating the major gene component to build either dominant, recessive, or codominant models. So in this case, the mixed codominant is very similar to the general model, but they've set the taus now equal to the Mendelian expectation. So they said, here in the general model, these were close enough to what we would expect. We're going to go ahead and set them now to their expectations so that we have fewer parameters to estimate. We're happy with these. Let's focus on the other parameters, which is essentially what's happening here. And then there's a purely major gene model where the heritability is set equal to 0. So now they're just saying that all of the genetic effects are due to the single major gene. There's no residual polygenic inheritance. It's a different type of model. This is, now what they're doing is just building dominant and recessive models where they're setting the heterozygote mean equal to one of the two homozygotes, which we said will give us a dominant or recessive model. And then there's a pure polygenic model where there's no major gene component, but now we're saying that all the genetic influences are purely polygenic. And then we have what's called a sporadic model, which is essentially one distribution. There's nothing genetic going on. There's no inheritance of that trait, just a single distribution in the population. And this is essentially just the mean, standard deviation, and then the likelihood. So those are the models. Most segregation analyses you're going to see will include models similar to these, but maybe different labels put on them. So now how do we actually go through and test and figure out what the best fitting model is? Well, we start over here with our general model. We say that this is the best fitting model. We look at our likelihood, and this has the best likelihood listed right here. This is the negative likelihood, so we want it to be closer to 0. And the likelihood here is 1890, which I'm sure you cannot read. But what we're going to do now is take that model as our model of interest and then compare the next nested model to that model. Now, what most people will do in a segregation analysis is the first thing they want to do is be able to reject the environmental model. You want to be able to say that that non-genetic model does not fit the data. And so what most people will do right off the bat is compare this, what they're calling a major environmental model, to the general model. And you can see here in this model, we've got a likelihood of, it's 1897, and we have 3 degrees of freedom because of the difference in the number of estimated parameters. And if we double that, because it's negative 2 times the log likelihoods for a chi-square value, this model is significantly different from the general model. So we say that this model provides a significantly worse fit to the data, and so we reject it, which is nice. Now we're saying the non-genetic model does not fit the data. Let's reject that. Okay. Then we go down to our next genetic model, which in this case we're calling a mixed codominant model. The only difference between this one and the general model is the taus are set. And essentially what we're doing here is saying, are these taus significantly different from what we estimated in the general model? Because those are the only parameters that are different between the two models, so that's what we're testing. So it's a model with 3 degrees of freedom. The likelihood is very similar to the general model. It is not significantly different. Okay? So we say that this model isn't significantly different from the general model. Therefore, we cannot reject this mixed model. What people then do with a segregation analysis, generally, is that this now becomes our best fitting model. It's more parsimonious, meaning we've estimated fewer parameters and it fits the data just as good as the more complex model. So now this becomes our favored model, and this is what we then compare the other models to. Yes?
What were the taus set to? I can't read it.
So yeah, the taus are set now to their Mendelian expectations of 1, 0.5, and 0. Okay? And so you can see then what we're saying is, because in the general model they were so close to the expectations, we're saying that this is not significantly different. We're saying these taus fit their Mendelian expectations, which is good. We like that. Okay? So now we're saying, okay, this is our general model. We're going to compare the other nested models to this one. And now the next model that is being compared is what we call the major gene model, which has no polygenic component. So now we're saying, okay, can we fit a model that just has the major gene component, and is that residual genetic component important or not? So that's the parameter that's being tested here. Now in this case, compared to this other model here, the only parameter that's set in the major gene model is the heritability. So we've got 1 degree of freedom. The parameter that's being tested is the heritability. And actually, the likelihood is significantly different. So we reject that major gene model, and we say, you know, we can't just fit the major gene. We have to have that polygenic component in there. And that's not surprising given how large the heritability was in the general model. It's not surprising that we can't exclude that parameter. So then we say, okay, that model is rejected. This is still our best fitting model, so now let's compare the recessive and dominant models to the codominant. So what's being tested in both the dominant and recessive models is a single parameter, which is the second mean. We're saying, is it either equal to the homozygote mu1 or the homozygote mu3? So it's a very similar test in both of those. And you can see, well, I will tell you, probably can't see, but in the recessive and dominant models, both of those models with 1 degree of freedom were rejected. So we're saying the dominant and recessive models do not fit better than the codominant, which again, if you look back to your general model, isn't surprising because these means we had 3 separate subdistributions. And we kind of guessed from that model that this might be codominant. Okay? And again, this is bearing out. It's consistent with what we expected given the general model. So both the recessive and dominant models are rejected. And then we go to a polygenic model, which essentially just has the heritability, and that was strongly, significantly different. So that, again, did not provide a better fit than our mixed codominant model, and we reject that one. Same thing with the sporadic model, which essentially says there's just a single distribution. It's not genetic, it's not environmental, it's just, a single distribution in the population. That model's rejected. So what we're left with from the segregation analysis is this mixed codominant model. Okay. So we would conclude that whatever this trait is appears to be influenced by both a single major gene that's inherited in a codominant fashion, as well as these residual polygenic effects. Okay. We get a little bit more information from this, such as the allele frequency. In this case, the allele frequency is estimated at about 0.83. So we would say that one allele is about 0.83, the second allele would be about 0.17, which again gives us useful information because we know for the less common allele, it's not that rare. An allele frequency of 17% for the less common allele is actually pretty high. Some of the things that we look at when we're looking at rare alleles are 5% or less. And so you can imagine to find any homozygotes in the population, you have to have a pretty large sample if that allele frequency is 0.05. To find homozygotes, it's 0.05 times 0.05, so it's 0.025 people who would have that homozygote rare genotype. So you can see as these alleles get rarer, to find the homozygotes, you need quite a large sample size. Okay. So this is at least reassuring. The rare allele is not too rare. So that's kind of the gist of what a segregation analysis is going to look like, what the parameters are, what the things are that you should look for. Now, as I said, I put this one up here as a nice example because in their general model, they were actually able to estimate all of these different parameters. As I said, it's not uncommon to not be able to estimate at least tau 1 and possibly tau 3. And so frequently, a general model has tau 1 and tau 3 set to the Mendelian expectations, and the only tau that they're estimating is tau 2. Okay? And that's not unusual. That's not a black mark against the paper. It's just typically what happens when you're trying to estimate a large number of these parameters. Okay. This was just a particularly nice example of a segregation analysis. So are there any questions on the segregation analyses? I mean, generally speaking, we have a limited number of parameters. You manipulate these parameters by either setting them equal to 0 or setting them equal to each other in different cases to develop these different models. And frequently there are many more models than what you see here, different combinations of things, and it kind of depends on what your data is telling you, which models you're actually going to build and test. Mm-hmm.
Well, this is very basic, but why is it called a segregation analysis?
You're essentially looking for co-segregation through the pedigrees of different phenotypes, and you're trying to model, are these things segregating in a way that you would expect if it was under Mendelian control. Yeah, there's some of these names, I have no idea how they came up with them, but these I know at least. Yeah.
So does the depth of the pedigree have an effect on how likely you are to be successful? And what I mean is by the number of generations.
Yeah. It's not only the depth of the pedigree, but it's also how large your sample is. As you can imagine, the more parameters you're trying to estimate, the more data you actually need to do that. Frequently, people have pedigrees where they're just nuclear families, so parents and offspring and no other types of relatives. Those you may be able to do a segregation analysis with, but sometimes it's difficult to get good estimates for some of these parameters. The bigger the pedigrees and the more depth, the more data you have to estimate these things. You'll also notice that these do not have any environmental covariates in them. There are models where you can include environmental covariates, you can include interactions, modeling gene-environment interactions, and for those types of analyses, you need a lot more data than just these basic set of parameters that you see here. Okay. Yeah. For segregation analysis, just to give you an idea of the sample size, it's not uncommon for people to have roughly 100 pedigrees in some cases, and pedigrees may range in size from 3 to 25, 30, or 40. Okay. Some of the pedigrees can be quite large. Complex segregation analysis can handle pedigrees of different sizes and different configurations. So you may have a quarter of your sample where you've just got trios, nuclear families, and then you may have another set of families that have very complex structures, large numbers of relatives in them. The one thing that is difficult for many of these approaches to handle is when you have inbreeding loops. In other words, you have inbreeding or line breeding if it's in animals, and it's difficult sometimes to deal with those types of pedigrees. Okay, so that's segregation analysis. As I said, you will probably never do one on your own, but at least if you come across them in the literature, you'll understand what you're looking at and appreciate the amount of time that went into doing those analyses. There are a variety of different packages out there that you can use to run segregation analysis. One of the most common ones is the Pedigree Analysis Package, or PAP, and then SAGE, which we've mentioned earlier. Both of these use maximum likelihood methods to estimate the parameters and determine the log likelihood. PAP is free. SAGE, as I said, you have to pay for, but it has a variety of different modules in it to run different types of analyses. We've kind of gone through the model testing already, where what we're doing is comparing the likelihood ratio statistic using the likelihoods from two different nested models. And then the degrees of freedom are equal to the number in the difference of estimated parameters. And we've talked a little bit about maximum likelihood methods, but here the likelihood of each model is proportional to the probability of the data given the model that you're building and the family structure. Now, in the example that we just looked at, all of our models were nested, and it was very easy to compare nested models and use the likelihood ratio test to determine what the best fitting model is. But sometimes you're in a situation where you're trying to compare models that are not nested. And you could imagine, for example, in that general model, if some of the parameters you were not able to estimate. In some cases, then, the models that you're most interested in comparing would not be nested. So there is another approach that we use to compare non-nested models, and it's called the AIC. Does anybody know what that is? Ike's information criteria, which is why we say AIC. And what we're doing there is using the likelihood to compare two different models. It's not a statistical test because the models are not nested. But if you're in a segregation analysis down to 2 or 3 models which all fit the data and you can't reject, in some cases you want to say, okay, of these, which one is the best fitting? You may use the AIC to help you decide which model you're going to select as the single most parsimonious model. And the AIC is just based on the likelihood of the 2 different models as well as the number of parameters that you're estimating. And what this does is the model that has the best likelihood and the fewest number of estimated parameters is the one that's going to fit the best. So this just helps you to pick out the most parsimonious model. But it's not a statistical test. Okay? And that, too, is something that you may encounter when you're looking at the results from segregation analyses. Because these models, as I said, are so difficult to build and so difficult to estimate all the parameters, in some cases your general model, especially if you've got a small data set, can be pretty limited. And then it's very difficult to have the other models be nested in that restricted general model. So once you get done with the segregation analysis, then you're kind of left with the interpretation of the results from all that model building and testing and all the other things that you've had to do. It's not just a matter of saying that of the models we built, we had a single major gene model or a model that at least had a single major gene component in it. You also have to reject the non-genetic models. Okay? If a non-genetic model fits the data as well as a genetic model, that would not be strong evidence that your trait is under genetic control. Okay? So not only do you want to accept one of the genetic models, you also want to reject the non-genetic model. Okay? As I said, as long as the genetic model that you accept has a single major gene component in it, that's good. If it's the pure polygenic model, well, that takes us back to the same issue we had before, that if your trait is truly polygenic, you may not have much of a chance of identifying any one of those single genes which contributes additively to the variation in the trait. Okay? A single major gene model is going to be the best. With these, you should always test the transmission probabilities. In the example we saw, they were able to estimate all 3 taus but in many cases, the best you can do is to estimate tau2. Okay? You should never, well, it would not be ideal to have your general model be the one where the taus are all set, all three of them are set to their Mendelian expectations. Okay? At a minimum, tau2 should be estimated. Okay? And together, these things protect you a little bit against false inference of a single major gene. As I said, many of the early studies that did not include a non-genetic model were really biased towards finding genetic influences because that was the only alternative to the general model were other genetic models. Okay? So should at least have a non-genetic model in there. One other issue which we are not going to go into in detail, but something that you should be aware of, is usually when people are selecting families for segregation analysis, they're selecting families in which the disease is segregating. It's highly unlikely that most of us would go out and be able to afford to do a population-based study where we randomly selected families and then look to see if a particular disease segregated in those families. Most of these families are ascertained because they have an individual with the disease that you're interested in. Okay? Most of the time, we go farther than that and we say not just a single individual in the family with the disease, but we want families, multiplex families, where there are many people in the family with the disease. You think about some of these early studies that we've done in breast cancer and prostate cancer and many of these other diseases, they have multiple affected individuals. And when you take families like this and try to estimate these parameters, particularly the allele frequencies, you might imagine that those estimates are going to be biased because what you've done is you've selected a portion of the population where that gene is segregating or is likely to be segregating. And so the allele frequencies can actually be overestimated. You can also, in many cases, incorrectly select the pick families where it's obvious that something is segregating, and so your mode of inheritance may not be correct either because you're picking the families where the gene or the trait is actually being expressed. And there may be other families where the gene is segregating, but it just is not penetrant. In other words, you don't see the expression of the trait. So with segregation analyses, there's this issue of needing to correct for ascertainment. Although I have to say it is very rarely done because it is difficult to figure out, in many cases, what the correct ascertainment correction is. So just be aware with your segregation analyses that some of the parameters can be overestimated, but generally if there's a single major gene segregating, a segregation analysis is going to pick it up, okay? Which I think that's the important piece of information from a segregation analysis. Like I said earlier, non-paternity, because we don't have genetic data necessarily, we're not able to actually test for this, but fortunately it doesn't seem to have a big impact on our ability to select models. Many segregation analysis packages can allow you to incorporate environmental effects and other covariate effects. PAP will do that. SAGE will too, which is nice. You can also consider adjusting for other known genetic factors if you know of them when you run these segregation analyses. So they can be as complex or as complicated as you like. We just looked at a pretty simple one. The couple of important assumptions here is that we are assuming etiologic homogeneity. We kind of alluded to that the other day. What we're assuming is that the underlying genetic cause of the disease, assuming there is one, is the same for all of the families. Okay, now that's obviously something we can't know up front, but again, this goes back to thinking about your phenotype, picking your families to be as homogeneous as you can. If there are clinical characteristics or other things that you think are important and might discriminate different forms of the disease, you should make sure that your families are as homogeneous as possible. One thing about segregation analysis is we generally do not estimate power for segregation analysis because we don't know what the correct genetic model is. So it's difficult to estimate power unless you have the structures of the pedigrees and you estimate it under a variety of different conditions. So it's a challenge. Okay. Sample size for segregation analysis, as we said earlier, the larger the pedigree, with more generations, generally better than smaller pedigrees with fewer individuals. Okay, so segregation analysis, we don't use genotypic data here. We're just relying on our phenotypic data and modeling. Can be very time consuming. This would not be the focus of a grant application. but again would provide you with preliminary evidence or supporting evidence that what you're looking at is genetically influenced, that perhaps a set of families that you have appear to be segregating the gene of interest. This would all be useful for in support of larger grant applications. So just quickly a few comments about collecting family data. Collecting family data is more time consuming and more expensive than just collecting cases and controls. Because larger extended pedigrees are more informative than smaller pedigrees, it's often important not just to collect the local relatives living within your area, but to actually collect all possible relatives who may be spread across the United States. Now, it's fairly simple to get blood samples from individuals and have it shipped to you on dry ice or something like that. But depending on what your phenotype is, it may be difficult to get the phenotypic information on family members who are not living in the same location. So that's a consideration when you're thinking about doing family studies. What type of data can you collect on everybody? The one issue that's become probably one of the limiting factors in collecting family data is the issue of confidentiality among family members and the concerns of many IRBs around collecting family data. My advice would be to work closely with your IRB if you're designing a family study and see what they're comfortable with, and it may take a lot of iterations. It's not as simple as it used to be, I'll just say that, to collect family data. Some of the major issues that the IRB may ask you about that you should think about ahead of time is publication of these pedigrees. If you're going to publish a pedigree, you need to think about a couple of things. If you identify information about the family that they're not aware of, such as non-paternities, how are you going to deal with that in your publication? Not just in the pedigree, but in the written description. So you need to think about that, because generally we exclude those people from our analyses. And so you have to think about how you're going to describe that. Other sensitive information that comes out, adoptions, abortions, other medical conditions, these are all pieces of information that come out when you're collecting this data that you need to be aware of, and think about how you're going to deal with it and maintain confidentiality, especially when you're dealing with families. General approaches to collecting this data, it's best to work with your IRB to see what they're most comfortable with and what they're going to allow. But frequently what we do is we contact the proband, and then in many cases the proband has to contact the rest of the family members, and then they contact you, which adds a lot of time to collecting the family data. Okay, that concludes our session today on segregation analyses. Okay, thank you.